So I guess it's a multidisciplinary audience today, but I guess all of us pretty much heard this term brain-computer interfaces. So what I would like to propose to you in my talk today, the overarching theme, is that I think the next revolution for brain-computer interfaces is not going to be invasive, but maybe controversially non-invasive. So I'll talk about that, and I think the main modality through which that will occur, principally, would be through the EEG. Now, I don't know if any of you have seen this website. It's quite new. It's called The Transmitter. I recommend that you check it out. It's about neuroscience news and opinions run by scientists and journalists. And just recently, I had a link that I thought was interesting because they just came out with these new results about trends in neuroscience. And what they found is that essentially if you look at the top trending term recently in the last 5 years, basically, EEG is the top trending term. So this came actually as a surprise to me, even though I work in EEG. But I could feel a little bit things are improving over the years. But I didn't think that it would be such a big increase, 5% from, let's say in the 1970s, EEG was big. You can see this bump here. And then, it went up actually consistently and increasing in gradient. So I think we know that EEG is now being used more in research and also outside of clinical departments, just like the remit of the epilepsy department. You can have EEG, in different clinics and so on. And here are the usual customers like Alzheimer's is obviously popular because of also the aging population, and it's something that we all worry about, dementia. And then you have also EEG as the second term here for most used term. fMRI is here on number 6. And in terms of machine learning and deep learning, also there are some recent trends. So I think EEG is on the rise, and the question is why? And I would also say that probably it's to do with BCIs. And the advantage is that in BCIs you need to have real time, right? And I come to the actual definition of what is a BCI. And this is by the BCI Society. They came up recently with an actual definition because there were a lot of questions about what is a BCI. But in this case, it's a brain-computer interface is a system that measures brain activity and converts it in nearly real time into functionally useful outputs, thereby changing the ongoing interactions between the brain and its external or internal environments. Okay, the two key things are here, that it measures brain activity that we understand. I mean, normal brain scans measure brain activity, but the difference is that it converts it in nearly real time, which brain scans don't sometimes do. So we need this temporal resolution, and EEG has that temporal resolution to make that happen. So just briefly to summarize in a nutshell, what is a BCI using these kind of definitions? It starts off with the brain and it's in a particular state that we measure. Okay, we measure either task-related brain activity or we measure spontaneous brain activity, doesn't matter. And then we have a sensing component, which is the neuroimaging, let's say an MRI, EEG, or intracranial recordings. There's some computation going on, which extracts some features from this activity in, let's say, real time. And then there's some version of feedback. There's always some feedback in a BCI because you need the individual to interact with the machine. So it's happening in kind of nearly real time, usually seconds. And this feedback can take different forms. It can be extrinsic, so it can be like brain stimulation, deep brain stimulation, or it could be some kind of non-invasive stuff like TMS, TDCS, or it can be intrinsic, which is like cognitive through actual neurofeedback that we call neurofeedback, classic neurofeedback. But what's key is that it's a closed loop. And so the output determines the input, and the input determines the output. That's the definition of a BCI. And from that, you can basically define it as a closed-loop brain state-dependent modulation. So you're trying to modulate brain states by giving feedback to the brain itself. And here's an example that I think most people were familiar with, DBS, but this is so-called deep closed-loop DBS. So let's say this is a study that was N equals 1 in Nature Medicine, where they applied closed-loop neuromodulation to one patient with treatment-resistant depression, and they had basically a sensor here in the amygdala, okay, and they record the activity, and then they stimulate depending on the state of the amygdala at that time. And here's the loop, for example, the machine learning loop. You see you have some ongoing neural recordings, and of course, if you're deep inside the brain, you can get very nice recordings very close to the neurons. You detect some biomarker, and then the neuron stimulator triggers some bursts of activity that will improve the neural network function, for example, in that patient. And what they found is that it actually correlates with gamma power in the amygdala, gamma power being a correlate of like metabolic activation. And if you, for example, stimulate this amygdala, you will then have a decrease in depression scale for that patient across time. Okay, but the question is, can we do this, firstly, non-invasively, and secondly, without brain stimulation? And that's also the focus of our lab, actually. We work with this so-called endogenous version of neurofeedback, which is where the brain is the controller rather than the machine. But the principle is the same. It's a closed loop. So you have some sensing here, let's say non-invasive EEG. You take it, you extract some features, like say alpha oscillations or some other type of EEG activity. You classify it as like higher or lower than some kind of threshold where you want to either promote or decrease this kind of pattern. And then you present a game to the person where they try to modulate this. And in this case, it's happening only through the person's feedback and cognitive control. So this has actually been demonstrated to work. It's been discovered in the 1960s, but it's been taken some time to come to the fore in terms of research, and finally there's some more research on this topic. Now, the question is what kind of clinical applications there could be for this. Recently, there's a collaboration we did with a group in Canada of Professor Rutlenius working with post-traumatic stress disorder, and we found that actually at baseline, if you take a group of PTSD patients on average, they will have actually decreased alpha power. So this is expressed by this blue decreased in the frontal areas. Okay, so they have essentially less alpha oscillations, which indicate a kind of a cortical disinhibition, sort of cortical excitation in the frontal areas, which fits with their symptoms of hyperarousal and lack of sleep, poor sleep. And then if you were to give neurofeedback to them, which in this study they did for like 20 sessions, and 20 patients actually, they reversed these abnormal lower alpha amplitude regions, like in the DMN region here. You can see this increase that wasn't present in the sham group. Sham being basically replaying somebody else's brain, not in real time. So they were actually receiving fake feedback. So that shows that there's some promise there, that we could deploy neurofeedback non-invasively to very large populations, not people who just get implanted with electrodes. And these are the results from the clinical trial. You can see here, this is the sham group, pre-neurofeedback. They both have very high PTSD scores. There is some placebo going on here with the decrease in scores, in PTSD scores over time, but this is far exceeded by the actual experimental group in neurofeedback, which were able to control the EEG. And as I said, that's coincident with increases in alpha, restoration of alpha activity, and the treatment effect size is about 0.75, which is a large effect size with a remission rate of 60%, with no patient dropouts, and comparable to any current treatment, pharmacological or otherwise. Okay, so what would be maybe the feature in terms of this kind of application? So you could have something like this kind of treatment, but not necessarily in the hospital, but even potentially somewhere outside, but scaling the EEG out of that kind of context of a lab or so on, or these are experimental sessions. In this case, you would maybe have some initial baseline data collection where, through a smartphone or through a portable EEG at home, you would record some EEG and some other peripheral biomarkers like ECG, GSR, and you would kind of determine, okay, where does that patient lie in terms of their abnormal brain activity? And how this, with machine learning and AI, how this relates to the symptoms. Then from that, you would basically devise a neurofeedback protocol which would do this loop where the person could train this activity in a certain direction. And technically, ideally, they would not do this in the clinic or at home or in the hospital, but they could do it at home. So you could track these measures even through wearables at home and have a more like a real-time, finger on the pulse in terms of what they feel and how they train. So this is kind of, let's say, the dream of something that could be like a future application. At the moment, it doesn't really happen. But one big, I think, drawback or one big obstacle to this dream can be basically encapsulated in one word, and that is noise. In all these situations, EEG is used, and EEG, by definition, is very noisy because the electrodes are not placed inside the brain. And so when you have electrodes inside the brain, like in DBS, the noise is very weak, and you have a very good signal-to-noise ratio. But when you have non-invasive EEG, basically you have a lot of noise. What I mean by that is, for example, here, this red trace. You can see this red trace is typified by a lot of this kind of large unusual activity. This would be actually muscle movement, muscle tension, that projects onto the electrodes, or for example, electrode movement you can see here. Okay? And of course, this kind of activities corrupt the actual signal, because once you're trying to transform these features into actual features that you want to classify, you will have misclassification, because your Fourier transform, whatever you're deriving from that, is going to be completely distorted by this noise. And the computer doesn't know, is it noise or is it brain? It's just getting that signal. So in the ideal case scenario, you would want something that magically removes this noise, right? To make it like a clean signal like this. So just recently in the section, we developed a tool that could try to do this and maybe achieve this in real time, because that's also key. You can take some time to remove noise. And we know EEG denoising is a laborious process. If you have enough EEG, you can select some parts that are clean. But in the case of brain-computer interfaces, every trial, every second is important. You don't have the benefit of time to select your nice little epochs. So let's say there's a metaphor here that I use for noise-canceling headphones for the EEG. So in some sense, EEG is like recording some kind of music from the brain, right? Just like when you listen to your headphones, your music. And then you might have some background activity that's contaminating that music. And that's essentially what we're trying to do. We're trying to separate the music from the noise, and we know headphones, fancy headphones, noise-canceling ones, they can do that. And that's what we want. You have some background signal, you have some noise signal, and you want some algorithm to try to separate those two out. And for example, in this case, GEDAI identifies this noise pattern and creates actually a matching anti-noise filter, just as if you would produce something like an anti-noise sound. But this is, of course, digital. And then this would be the cancellation process. It comes out just with clean activity and removes this noise from the signal, right? So this is the ideal scenario. And how does it actually work in terms of what's prior to this, and how is it different from what previous approaches have used? Now, you can use blind source of separation to separate noise and signal. And sometimes you need to classify this noise, for example, with ICA. You would look at the statistics of the signal. You need a lot of time to do that. You decorrelate the signals, and you find out, okay, these noises are kind of independent in statistic from the signal of the brain, and that's how we remove them. But we don't have enough time to do that here. So the other approach you could try is to actually use a physics-informed model by basically knowing something about the signal that you're trying to denoise. And what we know about the EEG signal that we're trying to denoise is that actually it's anatomically emerging from a certain area, which is the brain. So far there has been no real methods that have leveraged that aspect, where we know, okay, spatially, the signal should be coming from these intracranial areas rather than anywhere else from. So it's not a blind source separation method. It's actually a supervised source separation method. And the way it works is that, for example, here you have a so-called forward model, which actually models how brain activity propagates to the electrodes from these intracranial sources, right? Intracerebral sources. So this is a so-called forward model, and you can see these arrows pointing towards the electrodes from the inside of the skull. On the other hand, by definition, anything that isn't brain, it's outside of the brain, and especially external noises like muscle noise or blinks or electrical activity, they come from the outside. So they project differently onto the electrodes from physical space. And so you can leverage those two differences to actually try to denoise the signals by saying, okay, these signals come from the inside, and these signals come more from the outside. And that's essentially in a nutshell, this method. But you can, of course, look at the preprint for more technical details. So that's essentially in a nutshell what it does. And so this filter kind of throws away the components that are noise and keeps the clean signal. So obviously, it's a big claim for it to do that. And of course, just because something looks nice when you remove noise doesn't mean you've conserved the signal. So that's the main problem. How do you actually prove this kind of method works and actually contains the key activity of the brain? And CIBM has kindly also made a video about this, so I'm going to play that now. Hopefully it works. So there's a little demo about what it can do. But then we'll get into some of the validation afterwards on some of the data. Noises I already showed you, they're also labeled. So you see here there's an eye blink on one of the electrodes there. get to the point of how do we reconstruct or how do we validate this kind of method? Okay, we remove some noise, but maybe it's taking the brain signal with it. And so the way you can do this is try to actually define a ground truth. I mean, in real empirical data, it's very difficult to know what the ground truth is because the noise is mixed with the signal, and you don't know what is the original signal. So what we've done is we've done some ground truth simulations where we take some clean signals and we add on top the noise under different situations, and realistic noise that are actually recorded from the muscles in the eyes, and we project them onto electrodes synthetically and empirically and so on. It's all in the preprint if you want to read it, but essentially this video here will show you how this was done. So let's say you start off with some clean signal, and this would be here, ground truth. So this is clean EG, and then you can add some plus noise here. So you can see these noises that they're actually kind of independent of the signal, and then you try to reconstruct the ground truth from only the algorithm seeing this thing. Okay, so you can see now the remove noise selectively there. The bit that gets selectively removed. And so you can actually correlate this original signal, blue, with the GEDAI signal there on the bottom left, and see how much they correlate. So how much did these denoising algorithms recover this ground truth signal at the beginning? And that can give you an idea of whether you want to use it or not. Because if you remove too much signal, then that reconstruction is obviously going to be very poor. Okay? And actually, so we've released this code. Firstly, we've actually validated it, but we also released it for all the 4 main EEG toolboxes, so you can download it at this website for M&E, EEG Lab, FieldChip, and Brainstorm. And so the community can test this under different settings beyond the ones we have tested it for. But just one example of what we've done is, as I showed you, we've created these data sets with ground truth signal, and we ran different denoising algorithms that are kind of state of the art at the moment, what's used for EEG denoising, not really in real time, but they are still the standard that are used. So PCA-based tools that are based on variance, like large components that are noisy, get removed. IC-label and MARA, some of you will know, based on ICA, independent component analysis, which is kind of the gold standard for removal of independent components, and GEDAI, which is a generalized eigenvalue decomposition method, which you can read about in the preprint. And what we've done is we've created multiple data sets where we mixed in different artifact types, either ocular EOG, muscular EMG, noise, or their combination, so they're simultaneously present like in real life signals. Also with different types of temporal contamination levels, so 25 to 100%, so 100% being completely contaminated with no actual clean data available to calibrate the denoising with, which is the major problem usually. And GEDAI actually works with 100% contaminated data because it has this, theoretical model that it can use to search for the ground truth. And different levels of SNR, 0 dB being basically the same size signal as noise, and minus 9 would be 4 times larger noise than signal. Okay, and then we ran these kind of combinations of different versions of these data sets, mixed them together, and we created 6,400 data sets in one benchmark, which is basically related to 10 clean EEGs mixed with 10 different artifact types, 10 different artifact EEGs with 4 artifact types and so on. So this is the total benchmark. We ran all these denoising algorithms to that. And overall, across all of these different conditions, although in each condition, actually GEDAI was always the most performing, globally, GEDAI, you can see here, reconstructs the ground truth. And this is on empirical data, so it's actual data with different electrode locations from the ideal ones that you would have in a theoretical model, can reconstruct the original ground truth at 0.93 correlation coefficient. So that's significantly higher, almost 10% more than the best competitor so far. And it's also real time. For example, ICA, MARA, and IC-label, they're not real time. And that corresponds to a signal to noise ratio increase of about 9 compared to raw EEG, which would have no preprocessing. So you're starting at something like -1 SNR, and you're boosting this signal to 9. Okay? So that's a great recovery, and that's like under very large noise conditions, like 100% temporal contamination. Obviously, that's the simulation, but what we want is actual validation that this is useful for us as neuroscientists or clinicians, that actually this brain signal that we conserve is useful for treatment or clinical applications. And in this case, we actually put GEDAI through two types of tests, which are kind of decoding tests, where you try to decode some kind of ground truth from the data, if it's empirical, to find out whether you can predict this data better using this more clean signal. So in this case, for example, you have basically a task where the person simply has to say, did they see an Einstein face or a white square? Okay, it's a classification task. And there's a different brain response when it sees a target, so-called P300 response, than when it sees a non-target. Okay? And so this is an open data set that's available online. Anybody can do this kind of analysis with these noising algorithms, and using 73 subjects and more than 10,000 trials across all subjects. And essentially, you're doing a binary classification task. Did the person see Einstein, or did they see a white target? And you can say, okay, from the EEG data alone, what can we predict? Okay? And if you compare the raw data, so here raw, with the area under the curve classification of 0.52, it's pretty much random whether you can, from raw data, noisy data, predict whether they saw something. Because the ERP is very small, and the noise overpowers that biomarker, and you cannot anymore predict. So under a normal domestic setting, this would be impossible to do without any denoising. Now you have some of these offline ICA denoising tools. You can see those, they are automatic. Just like GEDAI, they don't do that much better than a random, but a little bit better. Then ASR is the PCA-based one, which currently is the standard for using real-time denoising at about 72%. And then GEDAI actually significantly larger at 0.8, which actually is the maximum classification rate that the authors used in their paper by throwing away some of the trials, bad trials, where in this analysis, we didn't throw away any bad trials whatsoever. So we achieved the same classification rate, even with keeping all the trials. Another example of decoding performance would be if you can predict, for example, the identity of a person by just looking at their EEG. Just like you would use a fingerprint on your finger to identify the person, you might have a recording of that person under session one time, one day, and then 5 years later, you might do another EEG, and you would try to see, can I find this person's identity amongst 100 other individuals? And that's this matrix here, correlation matrix, where you take a covariance matrix of each EEG, and you try to look for which one is it similar to. How does this correlation, is it maximal with one of these EEGs from the 100 others, right? And you can see this red line down the middle, basically telling you that with most people, their fingerprint identifies them because it's the maximum correlation with itself, right, across time. And then you can see, okay, if I denoise better or worse, can I identify that person based on their fingerprint? And you can see raw EEG gives you still very low identification accuracy at around 55%. And so is the case for these ICA denoising tools, which again are good in simulations, but they do not really conserve this neural signal that's necessary to identify a person through their brain activity. And ASR again does best, second best, and GEDAI is way ahead here at 90% for this type of data set. So that's very reassuring and very promising. And then the other question is, could you also use this for another types of BCI? So I mentioned the exogenous BCI. This is actually non-invasive BCI. So you use repetitive TMS to stimulate the brain under different conditions, and you can do it in a closed loop, the same way as you would do it in your feedback. But let's say a particular brain state, like let's say high alpha, then you stimulate, you try to reduce it, and then every time it increases, you stimulate again to keep it low. So it's again in a closed loop. But the main problem with this kind of thing, again, is noise. Because actually, to be able to record at the same time as you want to stimulate, you need to record the brain activity without noise. And when you stimulate with these large magnetic fields, electric fields like TACs, you inherently have a lot of artifact on the electrodes. And that's a major problem for implementing closed-loop, non-invasive brain stimulation at the moment. So we actually tested this. So here's the signal that you would see usually during TMS, repetitive TMS. You can see here these massive red lines. They're like 1,000 times larger than the background EEG when the stimulator is on. And from this, you would want to decode, actually, what's the underlying brain activity. But as you can see here, there's no way to see something in there because it's all mixed up. The other artifacts are blinks you can see here, and some movement, some EMG, but these ones are the main ones that you would want to use to capture the brain activity from. So actually, preliminarily, we tested GEDAI on this data set. And for example, here, you can see some of the cleaner data that is behind, that is GEDAI denoised. So if we just throw away the noise, this is what it would look like. And you can see that at the transition point, there's no major difference in the background EEG. Of course, this needs further validation, but there's also a potential for GEDAI to be used in this kind of application. So take-home message for today is that EEG is back due to a number of advantages, although it's been in the cold for a while. I think it's back, and I think we're pleased, especially the EEG section, that this is the case. It's 100 years, actually, from the discovery of EEG. At the moment, 2024 was the 100th centenary. Excellent, because it basically has excellent temporal resolution, I think, and that's its key advantage. It's also mobile, which you would need for brain-computer interfaces, and it's also affordable, cheap. And also it holds promise for scaling neurotech out of the lab, because there's new advances in denoising. One example is GEDAI, and the other ones are that signal classification is actually better now with AI and deep learning. So I think in the future, BCIs will be increasingly non-invasive, so we can go beyond the headlines of Elon Musk trying to implant electrodes in a few patients and go to actually scaling this up into larger populations, hopefully, with development of course, of wearable sensors and through deployment in clinics and domestic settings. Thank you very much. I thank my team and you for the attention.